Roughly half of all human cancers carry a fault in the same single gene. But here is the strange part. This gene is not a cancer gene. It is the cell's own defense against cancer, a tumor suppressor. Scientists named it the guardian of the genome, and for years, they were convinced it was the villain.
The gene is TP53. It sits on the short arm of chromosome 17. It builds a protein called P53, named, almost by accident, for how heavy it looked on a laboratory gel, about 53 kilodaltons. P53 is a master switch, a transcription factor that reaches into your DNA and turns other genes on. Its one responsibility: to decide whether a damaged cell is allowed to live.
It was found in 1979, but no one was looking for a cancer defender. They were studying a tumor virus. 6 laboratories at almost the same moment noticed a 53 kilodalton protein clinging to a viral protein called large T. Because it piled up inside cancer cells, they drew the obvious conclusion: this protein must cause cancer. They filed it away as an oncogene. They were wrong because of a beautiful trap. The very first P53 genes they cloned were broken copies. The healthy version did the opposite of what they expected. Put intact P53 back into cancer cells, and it stopped them from dividing. By the early 1990s, the picture had flipped completely. P53 was not an oncogene, but the most important tumor suppressor ever found. In 1992, David Lane gave it the name that stuck: the guardian of the genome. Here is what the guardian actually does. Every time a cell divides, it copies 3 billion letters of DNA. And copying makes mistakes. When P53 senses that damage, it slams on the brakes. It halts the cell cycle, holding the cell still while repair crews fix the error. But if the damage is too severe to mend, P53 makes a colder call. It orders the cell to destroy itself, a built-in self-destruct called apoptosis. A damaged cell that dies can never grow into a tumor. That is how one protein guards an entire genome. Now imagine being born with the guardian already half broken. That is Li-Fraumeni syndrome. Inherit a single faulty TP53 copy, and the lifetime risk of cancer climbs above 70%, often striking in childhood. And TP53 fails in an unusual way. Most tumor suppressor genes are deleted outright. TP53 is usually disabled by a single changed letter, right in the part of the protein that grips DNA. One tiny edit that blinds the guardian without removing it. Rebuilding a broken guardian is one of the hardest problems in cancer medicine. You cannot easily drug a function that has simply gone missing. The first attempt was audacious. In 2003, China approved Gendesign, a virus engineered to ferry healthy P53 back into tumor cells, the world's first approved gene therapy. Today, the frontier is drugs that snap a mutant P53 back into its correct shape, or that unleash the healthy P53 some tumors still quietly carry. From a protein on a virus in 1979 to the guardian of the genome, to the most mutated gene in all of cancer,
We spent decades learning what P53 protects us from. Now, we are learning how to protect it. The gene channel.